Hi, my name's Steve Taylor. I'm head of the Computational Biology Research Group at Oxford at the Weatherall Institute of Molecular Medicine, and also I'm Chief Scientific of Zegami. And I'm going to show you a few features of Zegami today. So Zegami is a way of visualizing thousands of images at once. So here we have 3,246 images, and each of these images are part of what we call a collection. Now in this collection we are looking at Xenopus, which is a frog which is translucent. So it means we can look at how genes are expressed in this particular frog at different stages of the embryo. So in Zegami it's like a big database, but you can see the way it works is over the web, and it's extremely fast to zoom in and out of. Now what it's really good at is taking a high level view of your data and looking to see for problems. And we can do this by targeting certain regions. So for example, here we have different metadata associated with this particular embryo. We have its stage 19, it's a particular gene which is hex. It has a concentration associated with it. Now this refers to the concentration of a blocking agent which blocks this gene's action, because what we're trying to do is block the genes and understand how they work by that method. So one thing we can do with all this information, we can do a simple search as you would do in any normal database. So VEGF is a gene, and as we look across here you can see all these look reasonably decent embryos, but what's really interesting is here we have this set of tweezers which has come through, and obviously we don't want that in the analysis. So what we can do in this case, we can, for example, say we want to keep all these. When I say keep, I mean we can tag these with this keep tag, and then you can see here that's saved all these as keep. Now if we don't want to keep that one, we can just turn it off there, and that's excluded it from our data set. So this is really useful when we're trying to clear up the data. And if you see any other weird ones around, we can do that as well. So another interesting way of looking at the data in Zegami is with the tags. We can do what's called a histogram view. First we sort by a property such as stage in this case, and then we can do a graph of it. So you can see here that this is how we can see a histogram of these various points, and we can see how they stack up, and we can also drill down to those and look at these at different levels. So that gives us a really nice way of being able to manipulate these data. This is the first part that we're going to discuss, and now we're going to look at part 2 in a second.